All right. Good afternoon, everybody, and welcome to the webinar of the African Society of Human Dental and Craniofacial Genetics. So today we're very pleased to have Professor Melvin Ambele on board, who's going to give us a talk. I would like to just read and introduce him, read a little bit of his bio and introduce him. He's an associate professor in the Department of Oral and Maxillofacial Pathology at the School of Dentistry, the University of Pretoria. He is a distinguished researcher with specialized expertise in translational research and focuses on the molecular genetics of oral and maxillofacial pathologies. Professor Ambele serves as an academic editor for PLOS ONE and as a guest editor for Frontiers in Genetics. In addition, he has been serving on review panels for the NRF and the SAMRC with regards to their research grant applications. Within the University of Pretoria, he is a key member of the School of Dentistry, PhD Committee, and the Faculty of Health Sciences Research Ethics Committee. He is also a member of several respected scientific organizations, including the African Society of Human Genetics, where we actually had the opportunity of meeting Professor Ambele face-to-face at the last meeting in Uganda, and the American Society of Human Genetics, the New York Academy of Sciences, and currently the African Society of Dental and Craniofacial Genetics. Professor Ambele, we actually look forward to listening to your talk today because it's certainly a topic of interest for us, and his talk is entitled From Genomic Landscapes to Clinical Innovations: Pioneering Diagnostics in Oral Cancer and Beyond. So thank you very much for joining us and for everybody on the call today. Professor, you can take it away, and we look forward to hearing you.
So thank you very much, Prof. Getty. And good afternoon, colleagues and members of the African Society for Dental and Craniofacial Genetics. And thank you for providing this platform to be able to share some of the work that we've done over the couple of years at the Department of Oral and Maxillofacial Pathology here at the University of Pretoria. So welcome yet again to our monthly webinar series. And just to inform everyone on the call, for those who haven't registered, it's good to please register for the African Society for Dental and Craniofacial Genetics. The registration is free this year, and you won't be paying any membership fee for this year. So today, basically, we'll take a look at some of the molecular genetic studies that we've done in oral cancer, extending to other oral pathologies like odontogenic keratocyst. Basically, to be able to understand the development of these conditions from a molecular genetic perspective in order to pave the way for discovering of new diagnostic markers. So to start with, oral cancer, as we all know, fall in the broad class of the head and neck squamous cell carcinoma, which arises from the squamous cells lining the mucosal surface of the head and neck region. So the head and neck cancer, as we know, it broadly encompasses the lip and oral cavity, salivary gland, the oral, nasal, and hypopharynx, the larynx. So it used to be the sixth most prevalent, cancer worldwide, but now statistics have shown that it's now the third most prevalent cancer worldwide and accounts for 7.6% of all cancers and 4.8% of all cancer-related deaths. So the risk factors that are actually associated with head and neck cancer, irrespective of the anatomical sites, are usually common, which basically ranges from smoking, which is tobacco, alcohol, environmental pollutants, and HPV infection. So at the extreme right, what you see is that most of the molecular studies that have been done on head and neck cancer, be it from any of the anatomical site, has largely focused on categorizing it into HPV positive and HPV negative. So we're going to speak to this later as to why the HPV status has largely informed studies or molecular studies of head and neck squamous cell carcinoma. So if you just looked here for the HPV positive compared to HPV negative, you will see that the oncogenes that are amplified are different. So if you see here, there's an amplification of different oncogenes compared to the HPV negative. The inactivated mutations of the tumor suppressor genes are also different in terms of the molecular phenotype for HPV positive and HPV negative, while some of the somatic mutations that have been reported are actually different between the HPV positive and HPV negative. So irrespective of the molecular phenotype that underline any of these tumor, the most important thing is that it all aids the cell to be able to achieve any of these phenotypic hallmarks we see of cancer, which basically either evading of growth suppressors, also enabling replicative immortality, allow the cells to divide continuously, activating invasion and metastasis, which is something that we know it's common with most cancer in terms of metastasis. Also inducing or accesses vasculature, because tumor cells have their own microenvironments that allow them to continue to divide and grow. So there is a need for blood vessel or blood supply. So tumor cells have been able to develop, their own vasculature for blood supply. Also to resist cell death, sustain proliferative signaling, and there are new other hallmarks which have now been added, which is avoiding immune destruction, tumor promoting infiltration, genomic instability and mutation, and the dysregulation of cellular metabolism. So these are some of the hallmarks of cancer, which, irrespective of the underlying molecular genotype or phenotype that you looked at, they largely fall into any of these categories. So moving as to why does HPV status actually matters when we look at head and neck squamous cell carcinoma. The reason being that even though the HPV status has a distinct molecular phenotype, the prognosis for HPV positive head and neck cancer is shown to be much better than that of the HPV negative. So they also respond better to treatments such as radiotherapy and chemotherapy. And what we know is that in terms of the diagnosis, they usually use the in situ hybridization for HPV DNA prop or immunohistochemistry for P16. As P16, it serves as a surrogate marker for HPV infection, which is something that has been broadly used for head and neck squamous cell carcinoma. But as we are going to see in one of our very first study that we looked at the molecular genetic studies of HPV expression and also P16 expression in oral squamous cell carcinoma. So here we look at a case of a 57-year-old male that presents with an ulcerative lesion, has a smoking history, was HIV negative, and the clinical diagnosis shows it was oral squamous cell carcinoma. So what you see here is the histological examination, which the pathologists that are on this call will greatly appreciate this picture that I'm presenting. So the top layer actually shows a moderately differentiated non-keratinized carcinoma, while the lower region shows a moderately differentiated keratinized squamous cell carcinoma. Here you have this intervening stroma that actually divides these two sets of non-keratinized and keratinized structure. So here is the P16 immunohistochemical staining of this tumor. The top layer shows as a P16 negative, and the bottom layer, which is heavily stained brown, shows as P16 positive. Like I've mentioned previously, P16 is used as a surrogate marker, largely, for head and neck squamous cell carcinoma. But in this case, HPV immunohistochemistry or the HPV in situ hybridization showed that both positive and negative sections were actually negative for HPV infection. So this shows that even though there's HPV positivity in this part of the tumor, it's still negative for HPV infection. So we then look at the molecular genetics of this tumor, basically using OncoScan. So OncoScan is a molecular technique that has been developed and specifically optimized for characterizing or looking at genetic aberrations in tumor that have been preserved within FFPE tissues. So what you see at the top here on the first panel, here it basically shows you, this is what we call the copy number state of the P16 negative tumor. So each of these blocks here show the different chromosomes. So it's from chromosome 1 to 22 on your X and Y axis. So the copy number state, which is a log2 ratio here, if it's 0, it means it's a copy number state of 2, which is what we expect for each cell, a diploid. But what you will see here are a series of lines, blue lines, that are actually up a copy number state of 2, or they are below a copy number state of 2. So if it's above, it means it's a gain or an amplification of that particular area of the genome. If it's below the line of 0, it means there is a deletion. So the lower panel of this P16 negative shows is the B allele frequency. So the B allele frequency is a measure of heterozygosity. So for each gene, we will expect 2 alleles, one from both parents. So this will give us a B allele frequency of 0.5, which is this middle region. However, if you see a split from the 0.5 region, it shows you there is a loss of heterozygosity. And some of this loss of heterozygosity actually results in inactivating mutations, usually on tumor suppressor gene. And that has been reported in many other studies to give rise to tumor. So if you look at this picture, you will see that there's a lot of genetic aberrations that are present with the P negative tumor. However, when you look at the P positive tumor, which is the panel below, it shows almost a clear karyotype. So it shows that the genetic alterations between the positive and the negative are actually quite different. So if we have to summarize all the genetic changes which we identified on the different chromosomal region, so here you will see this table actually shows you in the positive region, we have the amplification of genes that have been typically reported to be involved in other head and neck squamous cell carcinoma, such as your P63 and SOX2, which are the squamous lineage transcription factor. And you also have this oncogene, the PIK3CA, which is highly implicated in head and neck squamous cell carcinoma. You also have gain of function of some of this oncogene. So these are also present in the P negative tumor. However, with the P negative tumor, you will see there are more genetic alterations compared to the P positive, such as there is an amplification of genes that are involved in the RAS family, like you have your KRAS, you have your NRAS, and also there's EGFR. These are all oncogenes implicated in the development of this tumor. So what this basically shows is that the genomic profile between these two P positive and P negative are actually distinct. And when we look detailedly into the profile, what we saw was that the P negative tumor was actually mosaicism in most of these mutations. So what mosaicism means in this case is that you have different clones of tumor that harbors different mutations that are propagated differently in this particular region of the tumor. So Therefore, the molecular profile of the P negative epithelium is actually indicative of what is known as oral field cancerization due to exposure to tobacco. So the whole idea of field cancerization was actually first described by Slater et al. In the early 50s. So it's basically the presence of more than one independent area of malignancy in tumor adjacent epithelium in oral squamous cell carcinoma. And exposure to carcinogens was actually suggested as one of the main causes of these patches or field precursor lesions. So based on this molecular profile, we could be able to propose what could be a model for the development of the P negative lesions, which is basically due to tobacco exposure and insult of the normal epithelium. The P16 negative area actually acquire a mutation in the P53 gene, which has been reported to be highly involved in the formation of the field precursor lesion or patches. And this region could actually gain more mutation to actually evade immune surveillance or evade the cell cycle regulation. So they could actually gain any of these oncogenes or inactivating mutations of some of these tumor suppressor genes that will lead to the expanded fields replacing normal epithelium, which is what we see actually characterize the P16 negative area. And these could develop further to acquire many more mutations that could lead to invasive tumor. So therefore, in this study, we show that the expression of P16 in the presence of HPV infection indicates the existence of an alternative pathways for P16 expression, which basically does not depend on HPV infection. So that's one of the important take-home message. And the second being that the aspect of field cancerization should be highly considered when it comes to looking at the diagnosis and the development of oral cell squamous cell carcinoma. It is important to also consider the fact that there's an increased incidence of oral squamous cell carcinoma in young non-smoking adults. Yet there's no clear evidence to support the existence of any single determinant when it comes to this kind of cases. So that actually spurred up our interest to be able to look at one of these cases of a teenager with, or a tongue cancer with no risk factor. So this basically, it's just a demonstration to show the ulcerative lesions in the tongue of this individual with no risk factor. So there's no history of previous malignancy, nor any family history of head and neck squamous cell carcinoma. And the clinical diagnosis, again, shows it was a poorly differentiated keratinized squamous cell carcinoma. So we did the molecular genetic studies, again, looking at genetic aberrations across all the different chromosomal areas in the genome, and also looking at loss of heterozygosity on the different chromosomal areas. So what we basically show here is just a summary of some of the mutations that we did identified. So again, you have your typical culprits, which is your P63, SOX2, and the PIK3CA, which were both amplified. These are oncogenes. And also there were other oncogenes that we did identify. Of interest is the identification of a very significant somatic mutations on the P53 gene. I'm going to explain the significance of this mutation, this somatic mutation, as we go along. So firstly, we would like to report that none of the detectable mutations, such as these that I've indicated that are actually commonly mutated in head and neck squamous cell carcinoma, were identified in this case of a young adult with oral tongue cancer. So EGFR is highlighted here because in most of the studies that have been performed looking at young adults with oral squamous cell carcinoma, EGFR has been reported or has been implicated to be one of the main determinants. However, we see that in this case, that wasn't the case. So the most important discovery that we made was the detection of this high confidence somatic mutation on P53. So which is the first actually that was or has been reported in tongue squamous cell carcinoma. So what's the implication of this somatic mutation? It's basically the substitution of cytosine with thymine. So cytosine here is substituted with thymine at position 637. And that results to a stop codon. So it basically stops the translation of this gene to a functional protein. As a result of this mutation, no protein is produced. So therefore, there is no P53 protein. And we know the importance of P53 when it comes to the genome. It's like the guardian of the genome, as it protects cells that have accumulated mutations during cell cycle to be able to push aside for that to be corrected or be destroyed so that it doesn't accumulate more mutation and becomes tumorigenic. So this mutation is actually known to be present in individuals with Li-Fraumeni syndrome, which is high-risk genetic susceptibility for developing cancer. Also, however, we saw that there was a deletion in the GSTT1, which is a gene family that encodes for one of the glutathione transferase multigene family, which is important for eliminating metabolic carcinogenesis. So this may have contributed in the development of this cancer in this young adult, in a case where the individual is exposed to secondary or environmental smoke. So the question that we then ask is that could there be a potential biomarker for oral squamous cell carcinoma? The reason behind this question is to be able to look at the different genetic alterations that are present in individuals and to be able to see what is common and what is distinct. So we presented that in this publication. So one of the main drivers is that there's actually an absence of a definitive clinical biomarkers for any of the head and neck squamous cell carcinoma subtypes. So it doesn't matter what the subtype is. Definitive clinical biomarkers are actually not clearly defined. So that limits early diagnosis and also the monitoring of disease progression. So in these studies, we actually look at the clinically identical tumor. We also include the tumor adjacent dysplastic epithelium and also include the normal epithelium as a control. So we use one of the innovative or commonly used method now is the Valscope device, which is a handheld device that actually shines blue light. And when it's been shined in the oral cavity, the normal epithelia actually fluoresces, it's autofluoresces, it's green, and there's a loss of fluorescence in area that the cells are actually abnormal. So the goal is to be able to identify, again, the differences and the commonalities between the oral cancer, the tumor adjacent dysplastic epithelium, and the normal epithelium, which serves as a control. And all of these patients actually have a history of tobacco consumption. So this is just a picture showing one of the cases where you have, here's the tumor, here's the tumor adjacent dysplastic epithelia, and here's your normal epithelium. And using the handheld Valscope, you could be able to see the distinction between the tumor adjacent epithelia and the normal epithelium that autofluoresces. And this is basically just a hemiglossectomy section of one of the specimens that we looked at. So looking at the genetic profile of these different tumors, as what you will see as the different genetic aberrations on the different chromosomal arm of the different tumors from the different individual. So what we identified was that you could see there is a difference in the mutations on similar chromosomal arm across these 5 tumors. So what that tells us is there's a unique interpatient mutation profile for each of these tumors. We also identified a highly significant somatic mutation on the BRAF gene on tumor 2, while there was a somatic mutation also identified on tumor 5. So what is important for us to note here is that if you look at the mutational profile across the 5 tumor, you will see that of tumor 2 actually has a clear, genomic profile. So the level of chromosomal instability in the other tumors are actually higher than that of tumor 2. So what this is actually telling us is that tumor 2 could be what we classify as an M-class tumor. So this is a kind of tumor that is driven by a single somatic mutation rather than chromosomal instability like you will see on the other 4 tumors. So these are just some of the mutational profile that we did identify that were common to all of these tumor. Some have been reported in head and neck squamous cell carcinoma. Some have been reported specifically on oral squamous cell carcinoma. The ones highlighted in yellow are actually novel marker which we did identify in some of these tumors and also in the tumor adjacent dysplastic epithelia, as I'm going to show you later. So here you see is the tumor adjacent dysplastic epithelia of this 5 patients. So if you look at the tumor adjacent dysplastic epithelia of patient 2, you will see that you have more genetic aberrations here than you have in the tumor that I showed. So it's basically indicative that the clones that are present in this tumor adjacent dysplastic epithelia are actually different from that which is present in the tumor. As I've explained, that tumor is driven by a single somatic mutation. Here you find what is common among all these tumor adjacent dysplastic epithelia. It's an aberration on the GSTT1, again, which I've explained that it's actually susceptible to the development of cancer because it's involved in clear eliminating metabolic carcinogens compounds. If we look at the normal epithelium, what we found surprisingly was that there was a genetic aberration on the 3p21.3 arm. So this chromosomal loci has actually been reported in most cases in head and neck squamous cell carcinoma. However, the identification of genetic alteration in this chromosomal arm is not clear because it could either mean that this chromosomal region is actually either unstable or there's something happening. So this will need further studies to be able to look at known oral cancer patients, that is healthy individual, and look at these chromosomal regions to see if we do observe similar kind of mutational profile or not. So in this study, what we show is that we identified a highly significant somatic mutation on the BRAF gene, which is basically the substitution of a glycine by alanine at position 469. So this is the first to be reported in oral squamous cell carcinoma. While on tumor 5, we did identify a somatic mutation in the PIK3CA, which has also been previously reported in other cases of this cancer. The inactivating mutations that we found in chromosome 22q, 11.23, that chromosomal loci that involve the GST11 in all the TDAE regions could actually be preemptive of secondary exposure or an exposure due to carcinogenic compounds, since all these individuals have a history of tobacco smoking. We then ask an important question: is there a marker for progression in oral cancer tumorigenesis based on these studies? So for us to be able to identify or to be able to determine if there is a marker for progression is if we can be able to determine markers that are common in all the tumor adjacent dysplastic epithelia in these 5 individuals, and to be able to see that these markers actually progresses into the tumor. If we identify such a marker, then we could know that this actually signifies clonal expansion based on that particular mutational profile. So to our greatest surprise is that there's no molecular event that was identical or that was identified common to all the tumor adjacent dysplastic epithelia that actually progresses to the tumor to denote clonal expansion from a single progenitor clone. So what this suggests to us is that there's a high level of intertumoral clonal diversity. So basically, like I've shown previously in the mutational profile for the different tumors, you could see that actually each of these tumors has a different mutational profile. So that shows clonal diversity in terms of oral squamous cell carcinoma development in these 5 individuals. So, but what we did find that was common to the tumor is that there was an amplification in this region, the 3Q, the 5Q, 8Q, 8Q, 22.3, that involved, some of them are known genes, some of them are novel genes that have not been reported in all these 5 tumors. So this could actually indicate that there are potentially other markers that are possible to be included in the diagnosis of oral cancer if that could be validated in a large cohort study. But nevertheless, we also did identify what we call a putative oncogene, which was an activating mutation in this region, and also an inactivated mutations in a potentially tumor suppressor gene in the different cases of tumor adjacent dysplastic epithelia of patient 1 and patient 5, respectively. And what we found is that these mutations that were found in this tumor adjacent dysplastic epithelia actually progresses into the tumor. So in these different cases, we could say that this denotes a clonal expansion from a single progenitor clone in these different individuals. So moving on to what we have been focusing on beyond oral cancer is odontogenic keratocyst. So we've looked at this in both sporadic and syndromic patients, basically trying to understand the molecular profile and see if they could also identify some mutations that could assist us to be able to distinguish between the sporadic and the syndromic cases, or mutations that are common to both cases that we could use as a marker just for the diagnosis of OKC cases. So these findings, we've published it as well. And going into it, it's a brief background just to tell you that odontogenic keratocysts are actually developmental odontogenic cysts. So they commonly occur as single non-syndromic cysts or as multiple cysts in nevoid basal cell carcinoma syndrome. So the mutation in the PTCH gene is what has been commonly reported to be implicated in OKC's development, as up to 85% of this has been reported in the syndromic cases, and 84% mutation in this gene has been reported in sporadic cases. Also, there have been other studies that have looked at OKCs and have been able to identify other new or mutations in other genes outside of the PTCH gene, such as your CDKN2A, which is heavily implicated in other head and neck squamous cell carcinomas, your TP53, and a host of others. So our comparative genomic analysis between 4 sporadic and 3 syndromic cases of OKC was to be able to allow us to be able to see if we could identify some other unique markers. So here is basically the patient characteristics between the sporadic cases and the syndromic cases. So they both include male and female, and there are different positions in which the different sites, anatomical sites, for which the development of these OKCs occurs. So the genomic profiling of the sporadic cases you could see that there is an entire loss of heterozygosity on chromosome 9q that harbors the PTCH gene that has been implicated in the development of OKCs. So 3 of 4 cases actually shows this copy number loss of heterozygosity that involves this gene. You also see here there's a loss of heterozygosity on this 16p11.2 that was detected in the 2, 3, and 4, the cases. So moving to the syndromic cases, we find that there's also an entire loss of heterozygosity on the chromosome 9q arm that harbors the PTCH gene. So this actually confirms that this gene is heavily implicated in the development of all cases, irrespective of whether it's syndromic or non-syndromic. However, what is of interest is that we did also pick up, mutations, genetic mutations on 16p11.2 in both the syndromic and non-syndromic cases. So if I have to put them side by side for you to see, you clearly see that chromosome 9 was actually affected in both cases, as well as 16p. Also, there were other regions that were actually unique to the sporadic cases that were absent in your syndromic cases. So to these, therefore, we could say that genetic alterations in chromosome 9q that affect both the PTCH gene, which is the usual culprit, and the Notch1 gene that has not been reported in all cases, but has been implicated in the development of other head and neck squamous cell carcinoma, as well as the 16p11.2, which is new, that is now common, supports the role of other genes in the development of all cases, as well as the copy number loss of heterozygosity that we did observe in some of the cases that were only detected in sporadic OKCs and not in the syndromic cases may be useful for differentiating between these different OKCs. So what we could say from all these studies is that it is genetic and genomic studies are actually playing a key role in to be able to define the clinical phenotype from a molecular level. This can help us to be able to inform and identify new diagnostic markers, and also some of them can assist in monitoring the progression of disease and even treatment outcomes. So I would like to thank my team that I've been working with, team of experts that have actually been very, very talented to be able to provide all the support so that we could be able to achieve some of this work. That include Prof. Willy van Heerden, Prof. Maaike Pepe, Prof. Andre van Zyl, Dr. Liam Robinson, and Marlene. This team of expertise have actually played a key role in to be able to understand not only oral cancer, but also odontogenic keratocyst, not from a phenotypic level, but also from the molecular level. So thank you all.
Thank you very much, Melvin. That was very interesting talk, and I thoroughly enjoyed it. So we have a few minutes. Let me just look at the time. About 15 minutes for some questions, further discussions on the topics. So I invite you to please ask, or you can put them in the chat. Okay, I'm going to read out a question. It says, in young individuals, could the presence of OSCC be due to inheritance? What could be the place of using trios in such cases or trios?
Thank you for that question. Yes, it could be the case of inheritance. For example, the mutations that we identified, the somatic mutation on TP53, which is the first that has been reported, is actually inheritable. So this is one of the cases where it could be inheritance. But in the case of trials basically means that we could be able to pilot it in a larger cohort of these young individuals to be able to identify more molecular markers and see exactly what actually is the predominant predisposition factors.
So, I mean, in this particular patient, perhaps moving forward, you could involve the parents then? Or
Yes,
not
yes.
as much?
Actually, that could be something that could be done in this case to involve the parents and involve other siblings to be able to determine the pattern of inheritability of this particular mutation.
All right. Thank you so much for that, Melvin. Is there anybody else that has anything? You could just unmute and ask, or you could just raise your hand.
Yes, I have one question. So, how common is the mutation you are talking about for a bigger at the population level, and how impactful that for different parts of Africa?
Thank you very much for that question. So how big is the mutation is what we don't know, because we are the first to report it in this young adults. So we are still looking into the literature, but to be able to look at how prevalent it is or how common it is, then we'll be able to do studies larger than just what we're doing at a single site. And that should be something that we could do collaboratively with people from other part of the continent that are working in this field or has interest in this particular field.
Okay, thank you. If I may, so your cytogenetic findings, were they in, I mean, I know it's kind of, I don't think it's inappropriate, but what were the race of the participants? Because I want to know, was it present in like various races, or were all the participants one race? Given the diversity of
Thank you
our.
for that question. We actually did not report that, but the clinical record, it's something that one has to look at to be able to look at the race. But we didn't want to consider that initially. We just wanted to be as unbiased as possible and only cater for modifiable variables like tobacco smoking and all the others.
All right. Okay. Tell me, I mean, this is just a little bit of a logistics question. When you were doing your karyotypes, what mode of investigations did you use? Did you use any MLPAs or microarrays, fish? I mean, and just for my own interest, maybe others might be interested as well.
So we use the OncoScan is microarray based.
Mm-hmm.
Yeah, so it basically looks at all the genes that have been implicated or that are important in the development of cancer and also cancer-associated pathways, as well as some highly significant somatic mutations. So it's a microarray-based assay.
Right. Thank you. Are there any other questions that you would have for Prof. Ambele?
Yeah, one other question I have, that
Go ahead.
is there any report from US or anywhere else about this mutation? If not, what do you think this is the first finding?
Thank you. Are you referring to a specific mutations or all the mutations that I've reported on?
Oh, I was primarily very general because you said this is the first finding or something. Maybe you can choose or tell us whether maybe how much your finding is novel or never been identified before, or why.
Yes. If I can say they are novel. So like in the case of the GP53, it has not been reported in any of the head and neck squamous cell carcinoma. The same for the somatic mutation that we found on the BRAF. It has been reported in colorectal cancer and small lung node cancer, something like that, but not in any of the head and neck squamous cell carcinoma. So this is something that, we have to, to be able to, future studies should be able to look at this further in a larger cohort, and also employing some functional genomic studies to investigate some of the reported new mutations to find out what effect they have on any of the hallmarks of cancer that would actually pinpoint it to the development of oral cancer.
Okay.
Thank you.
Anybody else?
Yeah, I have a question. Thank you very much, Prof, for this wonderful lecture. While you were taking this lecture, you mentioned that there's a group of cancer that is driven by a single somatic mutation. You called it something I'm trying to recall. Please, can you just mention the name of the group of cancer?
So it's called the M class of cancer. So you have the C class, which is driven by chromosomal instability. So these are the common, cases that you find with all cancers. Like you have lots of mutations. When you look at whole, genomic studies or genetic studies, you find out there are a lot of mutations everywhere on the chromosomes. But for the M class, these are cancers that when you look at the genomic studies, the profile is actually very clean, but you find a single somatic mutation, which is actually responsible for driving that cancer. So that's what we mean by the M class.
Does that answer your question? Or do you want to ask anything further?
Yes, thank you very much, Prof. Yeah, I totally understand now. Thanks.
All right. Okay. I just want to quickly ask you, so how would, when you talk about identifying progression markers, what kind of progression markers? Because I mean, in terms of tumor progression, the way I understand it, it would be, yes, firstly, it would be multiplication of cells. Secondly, it would be perineural invasion, lymphovascular invasion, and all of that denotes or encompasses the term tumor progression. So I was just wondering if you could say or just highlight for me, what kind of tumor progression markers would you be interested in looking for?
So thank you for the question. So when we talk about tumor progression markers, we are not looking at any specific markers. So this is an unbiased studies, kind of you want to let the data inform you on what could be markers of progression. So markers of progression, in this case, we're talking about clonal expansion based on specific mutation. So if you look at your tumor adjacent dysplastic epithelia for all the different groups of patients, and you find out that there are some genetic markers that are common. And if you look at the tumor itself, and you also find these same markers that are common in addition to other markers, then you could link that to tumor progression based on those markers. And you could say those are the markers of progression because it progresses from your tumor adjacent dysplastic epithelia into your tumor itself, which could delineate clonal expansion.
All right. Thank you very much for that. Professor van Heerden, your hand is up.
Yeah, thank you. Ambele, congratulations. Very well done. Just to explain this last question, we basically didn't look for something specifically. It is look for what is possible. That is where the Wellscope came in. So the theory, easy speaking, is you will have a Wellscope negative area, which histologically will not be dysplastic. And then you'll have a Wellscope positive area, that may be dysplasia or already squamous carcinoma, and then out and out cancer as the three identified clinical areas that could be separated. And so the Bell-scope negative area that was not dysplastic, because some areas of Bell-scope negativity did display epithelial dysplasia, as we know can happen. So we looked at the areas that were Bell-scope negative without dysplasia, compare that to the, as Melvin showed, to the Bell-scope positive areas that were not malignant, and then to the third group, the malignancy. And if you could find something that persists there and accumulate some more mutations as one goes between the three scenarios, that would have been great. But as we know, the cancers don't read textbooks, and oral cancer is one of the most heterogeneous, as Alan said, cancers. So we couldn't establish that. And that also is a support. I mean, David Sadansky many years ago has looked at similar models like you had for colon cancer, where you can find the progression of specific mutations up to the development of malignancy. We couldn't do this in the case of oral cancer, which I think, as the case of Bell-scope, was a very nice model compared to the colon, where you have polyps and malignant polyps and then out and out cancer. Thank you.
Yes, thank you very much. No, that's certainly helped. So my understanding, given the heterogeneity of oral cancer, and this would be a South African study, what are your thoughts on what findings we might get if we explore, for example, West African populations compared to East African populations and Central? What are your thoughts? Do you think we might get different profiles, or do you think we might be very similar? Any thoughts on that?
So when it comes to first of all, the underlying basis about genetic studies, we have to acknowledge the fact that the population itself, our population, is genetically heterogeneous.
Yes.
To start with, and if it goes to other part of Africa, also we have different subpopulation. It's no longer African population, but different populations in Africa. So it's easier to say we will find different profiles, but I think the underlying basis about tumor development is that irrespective of how different this profile may be, there could still be an uncovering of potential panels of markers which would denote, the development of oral cancer, either in general or to a specific population.
Yes, because if you think of it, I'm just wondering if one does a similar study in various parts of Africa and then develop a panel, but I'm not sure at this point, would that inform management of that tumor if you get these different mutations? Perhaps we haven't gotten to that point as yet. But Melvin, you were at the Uganda conference, and I think you might have also heard the talk about HPV and oral cancer in HIV. Do you remember that talk?
Yes.
Now, their findings were markedly different to what I think we get here, where they suggested or told us that in oral cancer, in patients with HIV, they tended to be HPV positive, as opposed to here in South Africa, we have oral cancer that tends to be HPV negative, and the oropharyngeal cancer tends to be HPV positive. Do you remember any of that? And maybe you could sort of elaborate a little bit, because I thought that was quite or different to our findings, and I wondered why.
So I think one of the key messages that I got from that talk is that the HPV strain that they were looking at, it was different. So they spoke about different HPV strain, which is not the common one that we all looked for. So I think that was one of the highlights that he did mention. Yeah, I see Prof. Van Helder's hand up. Maybe he has something to say to that. Yeah.
Yeah, thank you, Melvin. I think one has to be very careful to extrapolate concept of HPV and head and neck cancer to outside the oropharynx. I mean, the second most common one after the oropharynx is sinonasal. We know 30% of oral cancers, will be P16 positive, but only 5% will be HPV related. And there's also no treatment beneficiary or benefit to the patient. Coming back to HIV, yes, we do, but there's a very specific type of oral cancer in HPV. We see it in these patients on patients who have AIDS, not on medication and so on, presenting with these bulbous, almost papillary-like squamous cell type of squamous cell carcinomas, and they are all HPV driven. But one shouldn't, in the HIV setting, we look at our number of patients, a lot of them smoke and they get the classical oral cancer as well. And then they don't behave differently or any other factors than to the one. Our HIV burden is so high that we have to look at specific clinical types of oral cancer. You cannot put it under one blanket and say, look, this is what the case may be. This is not our understanding from a clinical point of view. Thank you.
Thank you very much. I think that helps us understand a little bit better, especially here with our high HIV burden. Our time is up at this point, and Melvin, if there are any further questions, would you be open to people emailing you, or how do you feel about that?
Yeah, that's perfectly fine. I think my email was on I think they can just email me if they have any further question. I'm happy.
Thank you very much. Thank you, everybody, and thank you for joining. I know it's a Wednesday afternoon or Thursday afternoon. Sorry, apologies for that. And we will meet again next week. Thank you. Bye-bye.
Thank you. Thank you. Bye-bye.